So today I'm teaching about the different cell pathways and protein communication pathways in cells. So important terms to know for this from different points of learning in this unit. Phosphorylation is the hydrolysis of an ATP in order to attach the into ADP in order to attach the phosphate ion to a different molecule, and we'll get to what that means. And then enzymes are different proteins that have different functions. They act as catalysts, and they generally end in ACE. So, for example, first one that comes to mind is ATP synthase, which has that very typical ACE ending in it, and they just have different functions within the cells that we'll also get to. So, the first one we have to, or the first pathway we have is the G protein pathway. And so this protein pathway starts with GPCR, or the G protein linked receptor, or GPCR, which is the G protein coupled receptor instead of linked receptor. So just be aware of different names, XYZ. And an important thing to note about this one is it's actually a 7 helix. It has 7 helices in the structure. 1, 2, 3, 4, 5, 6, 7. So you see you have your bonding site right here. And so when you have a ligand come in here, it'll bond right here. And this is extracellular where it's bonding. And intracellular is inside the cell. So extra is outside the cell, intra is inside the cell. And so you have this 7 helix structure. So when a ligand comes and bonds in this site, then you have a conformational shape change within the G protein. And the G protein is made up of 3 subunits. And I'm not going to draw what looks like in its inactivated state because the active state looks different. And so because we have an active state right here after the conformational change, I'm going to specifically draw it in its activated state. But no, all 3 of these will be together in its active state. And so in its inactive state, all 3 will be together. So in its active state, you have the beta and gamma subunits, and they are together. And so this is beta, and this is gamma. And gamma specifically is the one on top because it's anchored as an anchor to the phospholipid bilayer. And so you have this gamma right here, you have the beta. Well, then you have a separate unit, which is your alpha unit. And the alpha separates from the other 2 subunits, and it performs all of your things you need done within the cell. So, or all of the pathways. And so your alpha, after its activated state, will come separately from the other 2. There's your alpha. And the first pathways we're going to go are the GI pathways and the GI pathway and the GS. So I'm going to start with GS because it's the most, GI will make sense with GS. So the alpha will go to a adenosine or adenyl cyclase. So I'm going to signify that with AC, adenyl cyclase. The adenyl cyclase will then take in ATP and create CAMP with that. It will change it into CAMP, which is a secondary messenger. And CAMP then becomes pKa, which is, this is named protein kinase A. And pKa is, we'll come back to pKa because it's specifically used in the tyrosine-kinase pathway as well, as signified by the K, kinase, protein kinase. And so this is the CAMP secondary messenger to create pKa. So this is the GS pathway specifically. So this is GI. The GI pathway is the exact opposite. GI inhibits all of this from happening. So the GI is basically the opposite of this pathway. And so what GI does is it then inhibits the AC from turning ATP into KMP, CAMP. So it halts this, and so it halts the production of pKa. And so these pathways are different and distinct and important both. However, there's also a third pathway involved. And the third pathway is the GQ pathway in the G protein. Okay, so the GK pathway is completely separate, and GK goes to phospholipase C. And this combines PIP2. This combines with PIP2. And what it does is it creates with this combination IP3 and DAG. And so IP3 you may recognize as a secondary messenger, and the long name is inositol triphosphate, as signified by the P3, right? And what IP3's job is to do is to release Ca2 plus from the Golgi apparatus, or rather the endoplasmic reticulum, And what Ca2 plus does is it carries out a lot of different functions within the cell, such as muscle contractions or secretion of spit, for example. And so GQ specifically releases this. And this pathway also still produces pKa it produces PKC, which is a different type of protein kinase. So this is protein kinase C, which is distinct. And so this pathway is also essential within the cell. And so when you have this ligand release from up here, and it goes back, in order to go back to the original state, remember you have what activated it all. The inactive state is bonded to a GDP. And so when it becomes activated, the conformational shape change, it phosphorylates this GDP into a GTP. So that activates this entire pathway. Well, to inactivate it, you have GT pace come behind and phosphorylate this back and remove it and turn it into GDP. And this turns back this into this inactive state where they're all together, and it halts the pathway or inactivates it. And so that's the G protein pathway specifically. So then you have the tyrosine-kinase pathway, which is similar but distinct. So in your tyrosine-kinase pathway, it's very distinct in that it becomes when bonded, it carries out dimerization, which I'll show you what that means. But first, I want to draw a phospholipid bilayer to show that this is in the phospholipid bilayer. So the tyrosine kinase pathway, the receptors for it are shaped like this through the phospholipid bilayer. And on the ends, they have tyrosine, which I'm going to represent with a T, but TYR is also correct for that. And so when a ligand comes and binds with them, it binds with both of these, and they both phosphorylize each other. And so what they do by doing that is they add a phosphate onto each of these. And so now each of these becomes a bonding domain, specifically a SH2 domain bonding site. And these are bonded by just different hormones and XYZ. And so that starts the whole thing. And a very distinct pathway in the tyrosine kinase pathway, and what it's kind of known for is these MAP kinase cascades, and kinase cascades are very specific to this. And so in this, it goes from, it has a, right here, it'll bond with a specific protein, and it goes from GTP to GDP, or rather, the opposite. I have that backwards. I'm so sorry. It goes from GDP to GTP and activates. And what happens now is that starts off this very specific kinase catalyst and kinase cascade rather, and RAS it takes RAS. This forms RAS in the process of this turning on, and RAS then turns into RAF, and from that, kinase, 3K kinase phosphorylates this into MEK rather. I don't know why I said Q. And 2 kinase, and this goes to ER. I was going to hit it myself. ERK, and this is then phosphorylized with just 1 kinase. It's an R. And what this pathway does, what this ERK then does, is it controls transcription within a cell and also proliferation. And so this is essential in the transcription of a cell. And all of that is found within the tyrosine kinase pathway. Tyrosine kinase pathway also is controlled by, or controls bonds with insulin to begin the process of letting in glucose into cells, and it has just a long list of different things. But I thought ERK was specifically important because of that transcription and proliferation of cells, and it seems very essential. Lastly, you have your ion channel receptors. So this is where we come back to that Ca2+. And so this CAMP, if we remember from earlier, CAMP releases IP3, which then signals for the cells for the endoplasmic reticulum to release Ca2+. So we come back to some of that. So your ion channel receptors are, just imagine this is a protein, or the phospholipid bilayer right here. So you have a protein in it. It's a channel protein, and it has a very specific shape right here. And so this is your allosteric site. Well, you actually have the ion. You have your ligands are going to come bond right here at the allosteric site, and this is going to turn into. It's going to cause a conformational shape change once more, and it's going to open up. And see that allosteric site has now changed the cell shape because of where it's bonded. And so now you have this pathway through here in which ions can be released or come into the cell. It's a really ugly arrow. And so ions can either leave or enter the cell this way, and namely Na+, K+, Cl-, and lastly that Ca2+ from right here. And this is essential in the neurons in your brain, and the neurons have this. So this is, the neurons are shaped like this and have this, and this is where these ions are stored right here. And this is how barbiturates and benzodiazepines work, because benzos and barbiturates allow for Cl to come in the cell. Well, so during this time, the inactive state is caused by a negative charge here, and the positive charge is active. So barbiturates and benzos, such as Xanax, Valium, work by allowing Cl to come to the cell, and they inactivate the cell. And so they open these ion channels specifically at the GABA receptor. GABA receptor. And the GABA A binding site. So you have these barbiturates and these barbiturates and the benzos come and bind at GABA, and GABA is considered an inhibitor, which means it's, again, that's why it's inactivating the cell, is it's inhibiting cell activity. And the barbiturates and the benzos will bond at this GABA and have it open for the Cl minus. And barbiturates increase number of the openings of GABA, so it will look like this. That was really bad drawing. So you have a very increased time between them. However, the benzos, no, I'm sorry, I've told you wrong. These are benzos. Barbiturates increase the amount of time the GABA is open. And so if a normal one looks like this, so this is a normal GABA inhibitor, or a normal GABA pathway. This is with barbiturates, and this is with benzodiazepines. And so that's how they inhibit the neuron activity within your brain. And this is running long, so I'm going to be very short with intercellular receptors. Generally, it's when you have fat-soluble, lipid-soluble hormones, they can come directly through, or you have small enough, so NO2 NO isn't a good example of one small enough, and they'll come in and they'll bond with a hormone inside right here, and they will come into the nucleus, and they generally affect transcription, is the general reason. And they create a hormone receptor complex right here, and they act as a transcription factor within your nucleus. And so that's how all these pathways work. Yeah, and personally, I think the ion-gated channels are the most fascinating, but I think all of them are very interesting. So thank you.